Bismillahirrahmanirrahim. Assalamu alaikum dear students, welcome to your channel The Anatomy Canvas. In Embryology Foundation module, today's class we are going to discuss the spermatogenesis. So first have a look on the male genital system. You know that the testes are present in the scrotal swellings, and this is the structure of the testes in which seminiferous tubules are present, and the process of spermatogenesis takes place over here. From here these mature spermatozoa are transported through the channel, this is the mediastinum testes and the rete testes, through efferent ductules into the epididymis, and then through epididymis they are transported through the vas deferens, and they enter through vas deferens and then into the urethra, and then they are ejaculated to the exterior through the penile area. So these are the prostate and the bulbourethral glands which add their secretion to the secretion of the epididymis. These are the accessory sex glands in the male, and this is the whole diagram showing the gross anatomy of the male genital system. A little bit about its histology, that the male gonad are the testes having 400 to 500 seminiferous tubules and per testes, and each having more than 0.5 meters in length. They are highly convoluted tubules present in the testes, and their length is more than 0.5 meter. Total tubular length is approximately 400 meter. This spermatogenesis process takes place over here. Then they are transported through the straight tubule into the channel called the rete testes, and through the rete testes they are transported through efferent ductules into the head of the epididymis. From here the conditioning of the sperms takes place, and after conditioning these sperms are transported through the body and tail of the spermatozoa into the vas deferens. In prenatal life, the spermatogonial cells or primordial germ cells are formed in a similar way as in the female genital system from the epiblast, and then they migrate from the epiblast by the process of gastrulation into the wall of the yolk sac, from where they migrate and enter into the area of the developing gonads. Here these cells are present a single cell surrounded by the follicular surrounded by the supporting cells, just like in the female genital system where supporting cells form the follicular cells. Here the supporting cells form the Sertoli cell. So at the time of puberty these testicular cords acquire lumen, and these cords become the seminiferous tubules. So under the effect of the LH, the process of the spermatogenesis starts at puberty. So here you can see at the basis the basically spermatogonia cells are present. These spermatogonia cells are type A spermatogonial cells and type A dark and type A pale. Type A dark spermatogonial cells repeatedly divide by the process of the mitosis and maintains the pool of the spermatogonia over here. Some of these spermatogonia type A cells then enter and become the spermatogonium type A dark cell, and then this start the process of the meiosis, and they first initially form the primary spermatocyte, and then primary spermatocytes starts meiosis 1, and after the completion of the meiosis 1 they form the secondary spermatocytes, and secondary spermatocyte after completing the process of meiosis 2 they form the spermatids, and then the spermatid transforms themselves into the mature spermatozoa. So duration totally is 74 days approximately in humans, and location is the seminiferous tubules where the process takes place, and also in the epididymis because maturation and conditioning of the spermatozoon take place in the epididymis. Purpose of the spermatogenesis is to preserve the number of the chromosomes in the offspring. So this is the whole story of the spermatogenesis. These are the testes covered by epididymis, where where the spermatogonia type A cell present, they form spermatogonia type B forming the spermatogonium. This is the spermatogonium type B, and spermatogonium type B then enlarge and become the primary spermatocytes. At this level you can see the cells are diploid. Here the first meiotic division starts and the two haploid cells are formed after first meiotic divisions, and then after this the second meiotic division starts and 4 haploid spermatids are formed. Then by process of the spermiogenesis, these spermatid transforms into mature sperms over here. So in this diagram you can see this is the primary spermatocytes having diploid number of the chromosome. Then it divides by first meiotic division into secondary spermatocytes, one having 23Y and other having 23X. These are haploid. Then they divide by the second meiotic division to form the 4 spermatids, and then by process of spermiogenesis these rounded spermatids will be converted into mature sperm cells. So stages of the spermatogenesis. First, two types of spermatogonia, as I have told you, type A dark and A pale, and then type B spermatogonia. Nearly 512 spermatids are formed from the single spermatogonium. During division, cells bound to each other by the cytoplasmic bridges for the synchronized development that is the incomplete cytogenesis. Here you can see from one spermatogonium a colon of the cells are formed, and all the colons of the cells are attached to each other by the cytoplasmic bridges, as well as they are embedded in the recesses of the Sertoli cells. So till the completion of the meiosis 2, these cells are connected to each other by the cytoplasmic bridges, and the process of the cytokinesis is incomplete. After this, the process of this, when they become mature and they form the mature spermatocytes, spermatocytogenesis is complete, after which spermiogenesis takes place, and in spermiogenesis the cells mature, after which they are released into the lumen of the seminiferous tubule. The process is called spermiation. From here they are carried to the epididymis, where they again become motile and their conditioning take place. The process called the maturation. So here is the process of the spermatogenesis. Type A dark, then type A pale, then type A pale into type B. Type B, after the process of meiosis, completion of meiosis, then enter into type B enter into spermatocyte phase, and spermatocyte phase completes the process of meiosis and form the spermatid phase. So then there are the different phases in spermatid phase. You can see first is the Golgi phase. In this Golgi phase, Golgi apparatus starts producing acrosomal vesicles. These acrosomal vesicles then coalesce, they form a cap-like structure to cover the nucleus, the second stage, which is called the cap stage. Then the third stage occurs, in which this acrosomal vesicles nearly completely covers the anterior part of the nucleus and this is called the acrosome phase. After this acrosome phase, condensation of the nucleus take place. At the same time, flagellum is also forming by the centriole, and most of the excess cytoplasm is extruded out of the body, which is engulfed by the Sertoli cells, and then it matures into it forms the mature motile spermatozoa. The process is maturation phase. So these are the different phases of the spermatogenesis. Here you can see this is the first phase, which is Golgi phase. Then the cap phase is started. Then the acrosome phase started. At the same time, extra residual amount of the cytoplasm is extruded. This residual amount will be engulfed by the Sertoli cells. Here you can see the principal piece, the middle piece, and the tail piece of the flagellum is forming, and the rounded spermatid is now transformed into motile and microscopic motile proper spermatozoon. So spermiogenesis is metamorphosis of small rounded spermatid cells into spermatozoa. After this process, the spermiation take place. During this spermiation process, the mature spermatozoa are then released from the recesses of the Sertoli cells into the lumen of the seminiferous tubules. From there, they are carried to the epididymis, where they again gain proper motility. Their conditioning take place, process called maturation. So this is the diagrammatic presentation of the spermatozoon. It is a haploid motile gamete cell having head, neck, body and tail, and these are the different pieces of the tail. This is the middle piece having the maximum number of mitochondria over here, then the principal piece, and then the tail piece. Exoneme, that is the microtubular structure in the tail that propels the sperm. It is having 9+2 structure of the microtubules. Acrosome, membrane bounded sac derived from the Golgi apparatus. This blue color area shows the acrosome. This is derived from the Golgi apparatus. It is having hydrolytic enzyme to help further in the process of the fertilization. Spiral arrangement of mitochondria over here. Speed is 1 to 4 millimeter per minute. Maximum lifespan of ejaculated sperm is 1 to 2 days. So these all are important from MCQ's point of view as well as also for your clinical knowledge, because for this point the fertilization should take place within 24 to 48 hours after ejaculation, otherwise these sperms will die. Also, their speed is important. It is 1 to 4 millimeter per minute, but at the time of ejaculation under effect of different hormones, also because the prostate and the bulbourethral gland secreting prostaglandin, which after ejaculation causes uterine motility, and under effect of these different factors, these sperms are sometimes carried within a minute to the sites of the fertilization. So there are different factors which are also affecting the motility of the sperms and carry them to the site of the fertilization as well. So this is the comparison. This is the female gamete. This is the oocyte, and it is covered by the zona pellucida and the surrounding cells forming the corona radiata. This is large immotile cell. This is a small microscopic having head, neck, and the tail piece. And here the nucleus is present, which is covered by acrosome. Small neck, which is connecting area. Then the middle piece, then the principal piece, and then the tail piece. So this is the process. The differences: primary oocyte alter DNA replication because 46 structure chromosome after tetrad formation they exchange their material. Here the primary spermatocytes after DNA replication, first meiotic division, second oocyte and first polar body is formed. Here after first meiotic division to secondary spermatocytes are formed. Here after the second meiotic division, one mature oocyte and another polar body is formed. Here at the end 4 spermatids are formed in this process. So these are the differences between the oogenesis and the spermatogenesis, important from your viva point of view as well as from your MCQs. So here are the characteristics: that the size of the cell oocyte is massive, sperm is microscopic. Then the motility: the sperm oocyte is immotile and sperm is motile. Sex chromosome constitution: that is oocyte has 23 eggs and the spermatocyte either 23 eggs and 23Y. Both the sperm chromosome constitutions are present. Start of gametogenesis takes place in prenatal life. Then there is a rest at the prophase in diplotin stage, sometimes up to many years, and at the time of the fertilization, then only few cells are selected and they complete their first meiotic division, and with each cycle few cells are selected in each cycles. And unlike to this in the spermatogenesis, the process completely takes place at puberty. So the number of the cells produced at the end is 1 mature oocyte, and here 4 mature spermatocytes are formed. Duration of the gametogenesis. Yes, it can take place up to 40 years because females' reproductive cycles nearly present up to the age of the 40. Sometimes menopause even takes place after at the age of the 50 years. Although some cycles near menopause are anovulatory, but in many women these cycles are ovulatory and the females are producing ova at the end also. So process which has taken place in prenatal life and completes at the age of 40 or 45, sometimes even 50 years. So it takes many years. In contrast, spermatogenesis at average take place nearly 74 days. So this is the whole comparison between oogenesis and spermatogenesis. Here I complete my today's class, and thank you. These are the references, that is Langman's Medical Embryology and the Developing Human by Keith L. Moore and the Prasad. And thank you very much, dear students, for your attention, and we will meet inshallah in the next class. Till then, Allah Hafiz.